This protocol provides a step-by-step procedure for sample collection, extract preparation, and immunoprecipitation for the confirmation of a successful protein pull-down and the detection of co-immunoprecipitated proteins of interest.
The protein extract preparation can be performed for up to 24 samples and is compatible with a number of downstream applications, including immunoprecipitation and RNA pull-down experiments. This method can be adapted to facilitate the testing of interactions between two or more endogenously tagged or overexpressed C. elegans proteins in a variety of genetic backgrounds.
Visual demonstration of this protocol is intended to make researchers comfortable with protein extract preparation and immunoprecipitation, hopefully encouraging those new to the techniques to use them in their research.
To collect a worm sample, first seed mixed sage or synchronized worms on solid nematode growth medium plates at the required temperature, and allow the worms to grow until the desired stage. At the end of the incubation, use M9 buffer to wash the worms into a 15 milliliter conical tube, and pellet the worms by centrifugation. At the end of the centrifugation, remove the supernatant, and wash the worms an additional 3 to 5 times in fresh M9 buffer per wash. When the supernatant is no longer cloudy, perform one final wash with double distilled water, and transfer the loose worm pellet into a 1.5 milliliter microcentrifuge tube. Pellet the worms with an additional centrifugation, and discard the remaining supernatant to obtain a packed worm pellet.